Hi, I'm Sashwat. I'm an MD PhD candidate in the Heimberger lab here in the Department of Neurosurgery at Northwestern Feinberg School of Medicine. In our single-cell sequencing of 21 samples that included high-grade gliomas, pilocytic astrocytomas, and normal brain controls, we found that the CAFs were associated with tumor grade, with high-grade tumors having more CAFs, and most of these CAFs were aligned with the immune modulatory subtype. Dr. Agi had defined 3 types of CAFs: the homeostatic, the mechanoresponsive, and immune modulatory. We found that it was mostly the immune modulatory in the high-grade tumors. We also started to look at what collagen types these CAFs were expressing within the tumor, and we found that it was mostly 6A1 and 6A2 within the tumor, and collagen 4 was a close second for this, and that was mostly associated with the vessels in the environment. Also, when we started to look at collagen receptors across the board, we found that layer 1 had a striking expression within the myelin compartment. Layer 1 has previously been described as an immunosuppressive protein, and so that really caught our eye as something that we can go after potentially in these tumors. There is currently a clinical trial for layer 1, which is something that's of interest to us now because there is an antagonistic for myeloid malignancies, and that could potentially be then with a new indication of gliomas enriched in collagen. We explored this on a pan-cancer level also, when we showed a similar association between Col6A and layer 1 across several cancer types, which means our study could be expanded to across several cancers.
Hello, I'm Dr. Hinda Najm, and I'm a postdoctoral fellow at the Heimberger lab within Malnati Brain Institute at Northwestern University. And as already my colleague Sashwat presented the transcriptomic data and findings of this paper, I'll be presenting to you the proteomic aspect of how we validated these findings. The COMET platform is what we use to validate our findings. It's a high-throughput, hyperplex workflow that is compatible with standard pathology tissues like frozen sections of FPE, where you can load 4 samples per run. It uses off-the-shelf unconjugated antibodies, and what it does, it's repeated cycles of staining, imaging, elution. At the end of the protocol, you generate omissive files that you can subsequently analyze using the viewer from Luna4, as well as bioinformatic analysis using standard bioinformatic softwares. It also allows the reuse of samples through other kind of analysis techniques. Key highlights of this platform: it is a fully automated system that you only need to load reagents, antibodies, and the slides. It is very reproducible, and it allows tissue preservation. And panel development is very flexible and rapid. So actually what we have available within the Malnati Brain Tumor Institute at Northwestern University is this pre-commercial unit. It's very similar to the commercial unit. It has 2 chambers. One chamber is for loading reagents from buffers to antibodies, etc. And another chamber is where you actually load the slides. It includes 4 stainers, and it has a microscope that is embedded inside. And the key technology of this platform is the microfluidic system that is actually based on this chip, where you load it on top of the slide, and actually everything else related to the protocol is totally automated. Most of the cell populations cannot be identified by one marker. Usually you need the co-expression of 2, 3, or even more markers at the same time to be able to identify certain populations. This is the example that we have for fibroblasts that were identified by Dr. Agi, and the ones that we are actually studying in this paper. So what I did is I generated for each marker separately the COMET images, and then what we did is we layered them together in one video to be able to make it more easier visualization of the co-expression of these multiple markers at the same time on the same cells. So in a high-grade pediatric glioma, I'm going to show you in this video how we are able to identify these fibroblasts embedded within the extracellular matrix. So what we are showing you in this video is collagen 6A in white, and the 4 markers that can identify fibroblasts: vimentin in purple, smooth muscle actin in orange, PDGFRB in yellow, and then PDGFRA in green. So what we are showing you embedded in extracellular matrix deposition, which is the collagen 6A, we can identify fibroblasts that are co-expressing all the different 4 markers highlighted by the different colors.
In conclusion, we found that pediatric samples did have the CAFs. These CAFs had a striking expression of collagen within the tumor. Also, they had the collagen tended to be associated with layer 1, which has been a previously underappreciated mechanism of immune suppression in gliomas and potentially also other cancers. Future studies will be directed at examining these layer 1 antagonists further in preclinical glioma models.